Hello and welcome to Sam Biology class. Today we shall discuss about two-dimensional gel electrophoresis. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Gel electrophoresis of proteins is most commonly performed in only one dimension. This technique is called one-dimensional gel electrophoresis. In this technique, after sample loading at the wells, when the current is switched on, proteins migrate towards the oppositely charged electrode at different velocity based on their size, shape and net charge. This migration results in the formation of lengths of protein bands. These bands can be compared easily with each other after staining or any other detection strategy. The intensity and thickness of bands are indicative of the relative abundance of proteins in various samples. The position of bands indicate the relative size of proteins. Although one-dimensional gel electrophoresis of proteins is most commonly carried out, this technique is not effective when proteins have similar physicochemical properties. This technique is also not suitable for the separation and fractionation of complex protein mixtures obtained from biological sample. All these problems can be overcome by a powerful technique called two-dimensional gel electrophoresis. Two-dimensional gel electrophoresis combines isoelectric focusing with SDS patch. Isoelectric focusing is performed in first dimension. It is a technique by which proteins are separated according to their isoelectric point. What is isoelectric point? The isoelectric point of a protein is the pH at which it carries no net charge. That means the protein is electrically neutral. We can say that at isoelectric pH, a protein carries equal number of positive and negative charges. The net charge of a protein is determined by the pH of the surrounding medium. If the pH of the medium is less than its isoelectric point, then the protein will have net positive charge and hence during electrophoresis it will migrate towards the cathode. On the other hand, if the pH of the medium is greater than its isoelectric point, then the proteins will have net negative charge. So during electrophoresis, it will migrate towards the positively charged electrode. But when the pH of the medium is equal to its isoelectric point, the protein will not move further and it will remain focused at that point as a sharp band. This is because at isoelectric points, different proteins are electrically neutral. In conventional gel electrophoresis, the pH between cathode and anode remains constant. So during one-dimensional gel electrophoresis, when biomolecules migrate from anode to cathode, their net charge remains same. By contrast, in isoelectric focusing, proteins are forced to move through a pH gradient. The pH gradient is stable and it ranges from pH 3 to pH 11. This pH gradient is created in a polyacrylamide gel by electrophoresing a solution of ampholytes. The isoelectric points of most of the proteins range from pH 3 to pH 11. In addition to ampholytes, the IEF gel also contains 8M urea, 8M urea and non-ionic detergents, non-ionic detergents, both of which will denature and maintain the solubility of the sample being analyzed. When a protein sample is electrophoresed through such a pH gradient, different proteins present in that sample will migrate towards the oppositely charged electrode. During this migration, proteins either lose or pick up protons. When all proteins in the sample are positively charged, then in response to the electric field, all of them will migrate towards the cathode. During this migration, their net charge is changed in response to the pH they encounter. During this movement towards the cathode, these positively charged proteins tend to lose more protons. As a result, they become progressively less positively charged. This migration of different proteins continue until they reach their respective isoelectric point, at which they become immobile and get focused into sharp and distant bands. In this example, we have 6 protein bands and each band has its specific isoelectric point. So by this technique, different proteins are separated according to their isoelectric point. After isoelectric focusing, the IEF gel strip is removed and is then incubated in a sample buffer containing sodium dodecyl sulfate SDS. After incubation, the gel strip is carefully taken out and is then placed horizontally on the top of a previously prepared 10% SDS patch gel. This is the gel strip which has been placed horizontally on the top of a previously prepared 10% patch gel. As the current is applied, SDS bound proteins will move from IEF gel strip into the SDS patch gel where they will be subjected to SDS polyacrylamide gel electrophoresis. Sodium dodecyl sulfate is an anionic detergent. It binds to proteins uniformly and suppresses their intrinsic native charge. As a result, all SDS-bound protein molecules have a similar charge to mass ratio. SDS-bound proteins are flexible and rod-shaped structure having a uniform negative charge per unit length. SDS treatment thus eliminates the effect of the differences in the net charge density and that of shape. The only difference between SDS bound proteins is their size. Because all SDS bound polypeptides are negatively charged, so during electrophoresis, all of them will migrate towards the anode. While migrating through the SDS polyacrylamide gel, all SDS-bound proteins will be subjected to the molecular sieving effect of the polyacrylamide gel matrix. Because of the sieving effect, smaller proteins will pass through the gel pores more quickly than the larger ones. As a result, smaller proteins will move faster through the polyacrylamide gel, whereas the larger ones will move slowly. Due to this size-dependent differential migration, proteins of different size will be separated into distinct bands. Friends, by two-dimensional gel electrophoresis, proteins are separated twice. In first dimension by isolatory focusing and in second by SDS polyacrylamide gel electrophoresis. Two-dimensional polyacrylamide gel electrophoresis has found extensive use in detecting changes in gene expression between two different biological states, for example, comparing normal and diseased tissue. Under favorable circumstances, up to 5,000 protein spots can be identified on a large format two-dimensional gel. Thus, with two-dimensional gel electrophoresis, we now have the ability to follow changes in the expression of a significant proportion of proteins in a cell or tissue type rather than just one or two. Friends, this is all about the principle, procedure and applications of two-dimensional gel electrophoresis. For more updates, subscribe my YouTube channel, Sam Biology Class.